So we believe in empowering young scientists with hands-on skills that can directly support their future research, employment, and postgraduate studies as well. So, without further ado, today's session is mainly focused on you guys to meet our speakers of the workshop. So, without further ado, I would like to go for the first speaker. So, the first speaker is Dr. Larinka Herat. So, he is an expertise in the synthetic biology and genetic engineering with a strong experience in both academia and industry. So as DMC, I think it's my duty to introduce you the academic background and the professional background of our speakers. So Dr. Lalinka is having a PhD in genetic engineering, slight size program between University of Colombo and Deakin University, Australia, and having a research mobility scholarship at Universidad de Granada, Spain, and also having MSc in Marine Molecular Genetics, Jeju National University, South Korea, and also pursuing the BSc in Molecular Biology and Biochemistry from the University of Colombo. So when we come for the professional roles of the Dr. Lalinka, he's a CEO, CSO of Biolume Islands, and also a Chief Scientific Officer at the Torrington Genomics and Bioinformatics, visiting lecturer at the University of Colombo and ANC Education, and also work as a researcher at the Center of Advanced Material and Device, University of Colombo, and also he worked as a former lecturer at NSBN and also while the University of Sri Lanka, also a former lead wet lab scientist, human genetic unit, Faculty of Medicine, University of Colombo, and also the application specialist, Haley's Life Sciences. So with all these academic and professional experiences, he has a research expertise in under several areas, mainly synthetic gene circuits, CRISPR engineering, genetic logic gates, genomic scale, design automation, computational modeling of biological systems. So as we all know, Dr. Lalinka is the one of speaker of the workshop. So I would like to briefly mention what are the main areas he will be covered. So he will give an introduction to synthetic biology and DBTL cycle, toggle switch design architecture, design SynBioHub and BioBrick retrieval, stimulating gene circuits, engineering logic circuits for therapeutic application, CAR-T-based conceptual modeling. So with this introduction to Dr. Lalinka, so dear sir, now I would like to warmly welcome you to address our participants.
Okay, thank you very much, Pabodi, for lovely introduction. Yeah, as Pabodi has given a great introduction to our company, Biolume Islands, as well as for our objective here, why we have gathered in this particular session. Yes, the Biolume Islands. So I'll briefly talk about Biolume Islands and then move on to what areas to cover in our first workshop that we brings you with Biolume Islands. Yeah, the Biolume Islands as a company, as you heard from Pabodi as well. So it is, we are recognized as Sri Lanka's the first synthetic biology-based initiative, and also we operate under this slogan of programming life, protecting tomorrow, which means basically our company is going to be working on projects where we are mainly focused on designing new biological organisms towards agricultural, environmental, as well as the health applications, which means the synthetic biology is an emerging subject area, a bit new to Sri Lankans, but it's getting the popularized among Sri Lankans as well. And so we will be want to guide and direct certain projects based on synthetic biology and to get you all involved as well in upcoming projects. So that's the basic idea. Why I will also quickly mention why and how Bioloom Islands originated as an initiative, because we saw the gap as a group of fresh graduates. So of course, among your some Sri Lankan fresh graduates came up with this idea that they realized that what we learn in the universities as well as in our higher education, uh, we learn a lot of fundamentals, and also we learn the conventional techniques in molecular biology, biotechnology related areas, and also other biological science topics. But we are really keeping up to date with what actually happens globally. So that question asked by your, like, some fresh graduates and also undergraduates. So we thought of creating a platform where we could all gather and then explore the real-time global knowledge, which can be integrated with our biotechnological research and also biomedical sciences researchers, right? So that's how basically it's not that something we wanted to forcefully create, but the idea, we saw the gap and we thought of integrating this platform for all of us to gather and explore these latest tools. So the workshop on applied computational biological tools. So this idea, again, as our first project, we thought of launching a workshop on computational biology tools. Why? Because we saw that as Sri Lankan scientists, Sri Lankan fresh graduates in this area, uh, we do not have kind of currently we do not have kind of idea how powerful these computational biological tools are, like for us, even for wet lab scientists, and also let's say there are scientists who are bioinformaticians and they are not seems to be working together. And also these are two isolated areas when it comes to Sri Lankan onset, but biology is biology, everybody. So biology cannot be separated with a margin that there's computational aspect as well as there's wet lab. So both wet lab scientists and also the computational scientists, they are trying to work towards a single goal to get an insights which are biologically sound. So our main idea here is to integrate this wet lab aspect as well as latest computational tool knowledge, and then see how we can uh, identify or define solution for biologically sound questions. So as the computational aspects are gaining and being developed really fast in rapid manner, we can't survive in the laboratory without knowing what and how powerful these computational tools are. So, as the first workshop, we thought of launching this workshop for us to understand, uh, gather together and understand how and why we have to utilize computational biological tools in our maybe ongoing research or any projects, and whether we can conduct research entirely based on computational and get answers for biologically sound questions. So that's to do that, we identified three main areas. Everybody, like whether we can use these computational tools to design biology or recreate biological system. So that's where I am going to conduct a session on uh, whether we can design biology to find answers for healthcare sector related research problems and also agricultural problems and environmental problems that we encounter. So to demonstrate this, if I narrow down to my session in this particular workshop, what I have picked as a topic is that I'm going to introduce you set of biological computational biology tools that we learn under synthetic biology as an emerging subject area in biology, and uh, whether we can utilize these concepts in synthetic biology to develop certain applied tools. We can answer questions in healthcare and also agriculture sector as well as in the environmental problems. So to do that, as Pabodi also introduced, I'll be initially letting you know what synthetic biology means, which means designing, reprogramming biological systems. So that aspect I'll briefly introduce in my session of on our Sunday session, and then I'll move on to what sort of biological in designing phrase, what sort of computational biological tools or software that we have to utilize. In designing phase, we will be mainly using one of the main tools that is being heavily utilized in synthetic biology, which is the SBOL designer tool. So that is listed in our outline as well. How to use SBOL designer tool? We will together go through the SBOL designer tool and see how we can actually design biological systems computationally with the going down to the genetic level. And then I move on to the simulation aspect. Not only designing, if we design something, we should be able to simulate and see whether these are actually working, the biological systems that you design computationally for health applications, environmental applications, as well as for the agricultural applications. To do that, we have a set of tools that we learn even under synthetic biology. One of the main tools is that iBiosim software. So we will be using iBiosim software uh, to simulate the small gene components that we design that can be integrated in biological systems. So those are the two main aspects. And there's one more database we need to access in synthetic biology session, which is known as SynBio Hub. So that's where all the genetic components are being stored that you can utilize in the designing phase of biological system. So we will be focusing on these three main tools, SynBio Hub as the database, and also SBOL Design as the tool that you can design actually biological systems utilizing that particular tool. And also you will be using iBiosim software to simulate a biological system. Towards the latter part of my session on Sunday, what I'm going to discuss with you is that, of course, we will learn how to utilize these tools in synthetic biology designing phase, computational designing phase, but what to design, how to conceptualize something biologically sound. Let's say you want to design a specific cell. Uh, or immune cells. We know immune cells can kill certain foreign material, attack certain foreign materials sent into our body, right? So because immune cells has this killing power. So how we can reprogram that ability of immune cells to target cancer cells developed in our body. So which means we are going to genetically reprogram a cancer immune cells, which are specifically known as CAR T immune cells. You can actually design a new CAR T cell to be targeted on the cancer cells being developed in our body. Specifically, how you can design a CAR T cell, how you can think under synthetic biology, how you can conceptualize a new cell that can kill cancer cells in the body in synthetic biology, utilizing synthetic biology concepts. So if I let you know, if I explain how you can think in designing a CAR T cell to kill cancer cells, based on that idea, you can start exploring how you can kind of broaden up your knowledge to apply in agriculture sector as well as in environmental sector. As an example, let's say there's microplastic contamination in a water body. So you are, as a biologist, as a scientist, you are asked to develop a tool, biological tool to degrade these microplastic and clean the water body. What you can basically do is that you can consider a microorganism uh, and you can genetically engineer that microorganism to capture microplastic out of contaminated water bodies and later to degrade it down to the components that are not no longer harmful or have adverse effects on biological organisms. So that's about environmental application. Even in plant reprogramming, you can apply this knowledge. You use recently we encountered this flood and our farms got affected. So how to create a genetically engineered plant that is resistant to flood condition and also drought condition. You can, of course, utilize these computational biological tools to computationally design a new intelligent plant that can adapt to flood conditions as well as drought conditions, maybe to resist for certain disease. So that conceptualizations can be done or designing can be done utilizing these computational biological tools that we are going to explore under smart gene circuits designing topic that I'll be conducting a session on in our workshop series, first workshop on computational biology. Yeah, so hope you got an idea about what we are going to basically do in this particular session. We can also further discuss if you have any doubts or anything to clear. I know this is a bit new area for you, but don't worry. We are going to simplify the concepts and start from the beginning and start exploring the basic tools and then to direct you towards which tools to be further learned if you want to design these advanced gene circuits towards multiple application. So idea is to share with you this knowledge in my session and for you all to integrate that knowledge in your ongoing research. Maybe you are working on your undergraduate research right now or you are planning your postgraduate research applications whether you can write research publications based on these aspects. So yeah, so those are the areas we are going to explore under my session in applied computational biology workshop on Sunday. So we will further discuss during our Q&A session as well. Yeah, thank you very much, everybody. Yeah.
Yeah. Thank you very much, sir. I think, yes, our participants have get a good idea what they will get through the workshop in this Sunday. So thank you very much, sir, once again. Then with that, yes, with that, and I have a quick reminder for everyone. So please stay until the Q&A session at the end that when we will share the special promotional code for the discounted registration fee. So keep that in your mind and you don't miss it. So then with that meeting with our first speaker, we can move to our second speaker for the workshop, in the upcoming workshop, is Dr. Samit Ratnayake. So I think it's my duty to introduce Dr. Samit Ratnayake. He is a computational chemist and a biophysicist whose work span molecular stimulation, computer aid, drug design, and data-driven modeling. He completed his chemistry honors degree at the College of Chemical Sciences Institute of Chemistry, Ceylon, and his PhD in computational chemistry at the University of Colombo. He has worked at Rajarata University of Sri Lanka as a lecturer and a senior lecturer in the Faculty of Applied Sciences and Faculty of Technology. He then held a postdoctoral research fellow position at the University of Missouri, St. Louis, USA, and a Horizon postdoctoral fellowship at the Concordia University in Montreal, Canada. His expertise includes atomistic and coarse-grained molecular dynamics, stimulation, rare event and enhanced sampling method, protein ligand system, peptide membrane system, molecular docking, QSAR, and Python-based cheminformatics and AI machine learning workflows, supported extensive experience with GPU, SLURM-based HPC systems. He is currently working as a scientist in the PlantFix Private Limited. So with this all academic and professional background, he has great research expertise in drug-target interaction modeling, structural prediction of macromolecules, molecular dissociation pathways, and binding site analysis. So as you all know, he will be our second speaker in the upcoming workshops. And during his workshop session, he will focus on accessing and filtering structural data from PDB and card-ready quality checks and structural alignment, file tracing and automation, integrating structural data into SBDD pipelines. So Dr. Samit's session is incredibly valuable for everyone interested in structural bioinformatics or drug design. So without further ado, Dr. Samit, yes sir, we now invite you to share a few words with our today's participants.
Hello, everyone. And thank you, Prabodhi, for that nice introduction. Okay. So in my session, in this upcoming workshop, I will mainly working on the computational drug designing. So in the workshop, I will giving you some kind of a hand-on experience of the very few steps of the computational drug designing workflows. So can I have the sharing option here? Okay. Okay, hope you all can see my screen. So in this upcoming workshop session, this is the the main things you can learn from this hand-on experience in this workshop. We will work with the Jupyter notebooks and probably in a local Jupyter notebook or the Google Colab, and use the protein data bank API to explore the metadata for a single PDB entry. And then we use this PDB search in API to find all the structure for given UniProt targets. Then we will form a Panda data frame to structure this the information from the PDB ID, title, method, the resolution, the release date, like metadata of that specific PDB ID. And then we apply some CAD-ready filters, like think about if you want to the filter out some specific X-ray ones, or whether the NMRO X-ray or the resolution cutoff, like if you prefer some kind of a resolution for the specific PDBI sets, and whether the ligand presence, something like that. So then we can select probably a good protein ligand complexes. So then visualize the selected PDBs in these Jupyter notebooks with the 3D images, like using Pi3 mol inside the notebook. And also, when you select this PDB set, we can identify ligand components IDs and fetch the ligand structure from the RSCB protein data banks. And specifically here we are using the RD-kit models to draw the ligand gallery from those selected PDBs and compute the basic descriptors from of these ligands like molecular weights, the C log P, HPD, HPA, and other rotational bonds and other very basic description we can compute from these selected ligands. So then apply the simple drug-like filters like Lipinski rule style things to filter out the ligands the furthermore. And so this is a kind of wrap up everything into a reasonable pipeline that can be run for any uniprot ID. So you all can adapt this into your own research. Okay, so actually this is a very first few steps of the actual computed drug designing workflow. So many people using the ready-made softwares to do these things, but the different from those ready-made software, this is a open source and Python. Then you can tailor these as your interest. You can adapt to your own research, not only for the computational drug designing, for the other ones. Apart from that, I just need to mention that I think if you are registered for the workshop so far, I think you will get a preparation guide. So this is kind of an essential thing to if you are participate for this upcoming workshop, because we are working in a Jupyter notebook. So certain things you need to preparation before the workshop. So as I mentioned, we are working as Google Colab or the local Jupyter notebooks. So I think one day before the workshop, we can send the GitHub link to this Jupyter notebooks, which is we are using in the workshop, and maybe the as a file, IFI file via email or shared drive. And also you just need to these things are essential when you are working, participate for this my session. You need to have the laptop and the stable internet connection, and probably I'm recommending you need to get the Colab account. If you have a Google account, you can easily do that. Or when we send the Jupyter notebook, you just need to open in your local machine or the laptop when it is going in the workshop. And yeah, so here in the preparation guide, you can see these the main things, and you just need to install these basic Python codes to in these Jupyter cells to start these things. Otherwise, when we start the workshop in the 8 a.m. in the morning, little bit, if you're not pre-prepared for these things, you will be getting a little bit trouble, because we are in a very tight 2-hour time schedule. So we need to go one by one in the those steps. So for that, you just need to install these pandas and the other modules. So yeah, so most of the guidelines are in the document, And also we will give you the Discord support. Hope you are, if you are already registered, hope you are already added to the Discord also. I will show the Discord also. So there we can help you with your problem with the installation and the other things. If you have any question, you can put here, and maybe you can use a screenshot to put there, and we can help you for installation and the other technical things. Okay, so yeah, so those are the things I just need to mention before the workshop. So yeah, that's all I just need to talk. And Prabodhi, yeah.
Yes, thank you very much, sir. I think our participants get a good idea, but they have to prepare before the workshop and how they have to make the plan before all of that. So thank you very much, sir, once again. So with that, we can meet our third speaker for the upcoming session. So our third speaker is Mr. Kajan Muniswaran. So Mr. Kajan is a molecular diagnostic researcher with extensive hands-on experiences. So when we talk about Mr. Kajan's academic background, he is a current PhD student in molecular biology, University of Colombo, and also academic foundation in molecular biology and information technology, and additional training in the Institute of Personnel Management as well. When we talk about his expertise, he is well expertise in PCR, RT-PCR, HRM, and gene expression analysis, cloning and sequencing, DNA RNA extraction, and also molecular genetic diagnostic as well. So when we talk about Mr. Kajan's professional experiences, he's a researcher, Department of Chemistry, University of Colombo, and also he was a former managing director, Cryptogenetics Private Limited, and also he was a research assistant demonstrator in several departments at University of Colombo. So his research focuses are under molecular diagnostics for health conditions, PCOS-related molecular markers, FTO gene, and anticyclic induced weight gain. So as he is the third speaker of the upcoming workshop, in his session, he will cover qPCR fundamentals, HRM assay setups, variant retrieval, and SNP interpretation, primer designing tools, qPCR optimization, introduction to genetic association studies. So this module is perfect for anyone in molecular genetics and diagnostics. So, Mr. Kajan, so we warmly invite you to greet and meet our participants and share your thoughts.
Thank you, Prabodhi. So you have heard of most diseases, and what is the most recent pandemic we had is the COVID-19. So what we are talking about is there are many variants we have came across. You may heard about alpha, beta, delta, and most probably Omicron. And also there are something in common with all diseases most of the time. When there is a disease or widespread in diseases or inherited disorders, even some other cancer things, one thing in common, that is the mutation. So in mutation, generally there are several types of mutation. It can be a single base pair mutation. That is the very uncommon thing, but sometimes it's more valuable and it's more promising to cause a disease. So if you came across this factor V Leiden, it is the mutation that causes the deep vein thrombosis. So people with this single nucleotide polymorphism, they will get the thrombosis. They will get the blood clot risk higher. And also they can get cardiac problems and all due to this blood clotting issues. Also, you have heard about this MTHFR mutation that is called C677T. So that is the causative mutation for this folate deficiency. So these are the mutations. Also, you can find economically valuable breeds of plants and also animals using the SNPs. And also you can monitor these viral strains and also the pathogens, how these spread, how the mutations evolve, and how it decides the virulence. Those things you can decide by this mutation. So knowing whether the mutation is in their genome or not is most important thing in molecular diagnostics. So one thing, and also for this lower middle income countries like Sri Lanka, we have a problem with this cost when if we are going for the NGS or for the high-end scientific techniques, the cost and switching cost and the reagent cost, everything is unbearable. And also the poor people in the country that they cannot afford. So these are the major issues we are coming across. So one solution is that we can go for this high resolution melting analysis. After the COVID, most of the laboratories having this qPCR instrument, quantitative real-time PCR. Using that, you can genotype most of the variants uh, using the probe-based methods. Even the probes with this fluorescent molecular bit high in cost, and also for the large scale, the cost is a typical thing, critical. So you should manage the cost also, and also you want to get some promising results, the endpoint PCR results that are not much accurate enough. So that's why we move to the quantitative real-time PCR. So in the quantitative real-time PCR, you can identify the mutations in a closed tube method. You cannot touch the samples sometimes because they can affect your health. So in this condition, you should need a closed tube method. You cannot do the PCR and load it into a gel and run these assays. So that's why this qPCR instrument is most critical. That's why with this COVID pandemic also, the government of Sri Lanka also purchased most of the qPCR instruments. So when I come to high resolution melting, so this DNA having some property called this melting temperature. The melting temperature decides how the DNA could be distinguished. The melting temperature we can monitor, but qPCR instruments having a small accuracy, about 0.2 degrees Celsius. So there are some other techniques we are going to discuss in this lesson, and also you will get the hands-on experience on how to design the primers and also how to design your own assays for any type of variant, including the SNPs and also insertion deletion polymorphisms that are called indels, and also for some structural variants such as CNVs and also for Williams syndrome, those things you can learn in this lesson. So that's it. So we will join again in the Q&A session. If you have any questions, you can feel free to raise your questions. And also we are ready to help you. And also we hope we will see you everyone in this Sunday for the workshop. So thank you very much.
Thank you very much, sir, for giving that introduction for the upcoming workshop regarding the molecular side. So then with that, we have come to the Q&A session. So I told you in the beginning, so it's a kind of a very important session that you can clarify a lot of things from our speakers, and also we are going to share a special promotional code. So I think our member will share that one. So with that, we will now open the floor for questions from our participants. So I know all the participants have a good idea with our speakers' thoughts, what they are going to get in the upcoming workshop. But still, you may have some questions about maybe about the workshop content, tools covered, or the career pathways, research opportunities, or either internship opportunities at the Bioloom Islands. So anything else, if you are curious about, you can ask from our speakers. So please feel free to raise hand and or type your question in the chat or whatever. You can unmute your microphones and ask the question from our speakers. So I think all of our three speakers are ready to clarify your questions or anything that you want to clarify from them. So dear participants, please speak up or type your questions in the chat box.
Good evening, everyone. I think I am audible, right?
Yes, you're audible.
So, first of all, I need thank you so much for all the speakers for delivering an amazing explanation. So, my question is, as an undergraduate student, how can we integrate this technology or the tools that all of the speakers have named us for undergraduate research work and for to join us? So can I get more information about it as well? Thank you.
Dear sirs, I think it's a bit difficult to hear, but I think you got an idea what he was asking about. So, Dr. Lalinka, Dr. Samit, and Mr. Kajan, could you please clarify his problem?
Brody, can you repeat the question?
Yeah, so I think he was asking about how what is the way that they should integrate this area with their undergraduate research fields. I think I could hear like that.
Okay. So actually, these three sessions are bit different to with different domains, but in a one kind of a main computational biology domain. But under that, for the main domain, these three are with different sections. So if someone interesting, yeah, if I talking about my field, if someone interested about to going through their undergraduate research related to computer-aided drug designing or some structural biology things, this is a very good chance to get some some kind of an idea how the computer-aided drug designing workflows are we can implement what are the very first steps of the computer-aided drug designing workflow, how the PDB data banks, data, UniProt data, how we can integrate those data into our workflows, how to separate protein ligands in automated. Specifically in my section, this is a very open source and also the automated one. So you no need to do it one by one with the manual things to separate your every PDB file treatment. From this kind of automated Python workflow, you can do thousands of molecules at once. So yeah, I think the Lalink and the Kajan also can give some answer related to their sections.
Yeah. Kajan, do you want to go next to elaborate more on why the HRM matters?
Yeah.
Right? Yeah.
So, as I told you, this high resolution melting based SNP genotyping is most practical thing, and we have optimized it for even the nearest neighbor neutral mutations. That is very hard to detect in the ordinary qPCR instrument. So we have experimented, and we had some promising results, and we can apply to N mutations even it's a plant or virus or a pathogen or a human, you can apply to anything. So you have to develop an assay, and also you should have a compatible qPCR machine, and also you need this saturating dye-based chemistry, the qPCR reagent. If you're having these things, you can practice this in any qPCR lab. So that is the practical aspect of this high resolution method analysis. So
Okay, yeah,
that's it.
to speak a bit about my area as well. So I know for undergraduates in Sri Lanka, the synthetic biology is a kind of a pretty much a bit new area for you all to explore. Here, what we focus on the designing aspect of biological system. Um, Not much explored uh, during throughout undergraduate research conducted in Sri Lanka, but currently working with multiple institutes, Sri Lankan institutes. Some of my students are also working as undergraduate to how to understand and to explore how we can utilize these design principles in biology to develop new maybe microorganisms, let's say like to design new plants and also to design maybe the human cells, how you can reprogram human cells toward finding new cures. So likewise, you have broad range of areas to explore even as undergraduate students when it comes to synthetic biology research. Now, if I take an example, let's say it doesn't matter that if you are currently working on a conventional topic, if someone is, I'll start with some basic idea that if someone is currently working on utilizing a microorganism, a wild type organism or the naturally existing organism, maybe to degrade the environmental pollutants. So there's a traditional approach in microbiological workflows that you culture microorganisms and look for the special strains that exist in the nature that can degrade these environmental pollutants, and then you are trying to isolate them in the laboratory. But idea here in designing aspect is that what we basically do is we utilize very well annotated biologically important microorganisms like E. coli strain. And then we are trying to genetically engineer that particular bacteria to give new characteristics for these bacteria to get the capability of degrading this particular environmental pollutant, which means you are trying to develop a new organism that can be utilized in such environmental application. So these designing aspects doesn't matter whether you are an undergraduate or you are just seeing you are a postgraduate or maybe you are conducting a research in during your mid in your PhD project. So you can think of integrating a designing component as well as sideline work on your conventional aspect that you are currently been working on maybe. So that's what I was like suggesting as well when I joined one of the institutes. So what happened, I was given a set of students who are working on conventional projects. Still, I expanded their projects by introducing another component for them to write a new chapter in their thesis. To design a new organism as well. So you can maybe combine both your wet lab work plus try to introduce a new chapter on designing new organism. Maybe to illustrate how you can later using wet lab technique later to develop that organism actually. So likewise, so that's related to bacterial microbiology application. So you can still explore health-related human application, and there are interesting techniques in designing organism that can be applied in plant-related applications as well. A lot to explore when it comes to even for the undergraduates here, everybody. Yeah.
Thank you very Right. So I think we have some other questions in the chat box. So we will start from the first one. So the first one from Dishani, she was asking about if we need our future practices and learning in those mentioned things, do you provide chances and anything by your side?
Sorry. Then I'm Dishani Pramodya. Then
Yeah, sure, designing. So you can ask the question, please.
I'm working as a teacher lecturer, and I was really emphasized, and I really very thankful for the inside knowledge sharing. Those three resource persons actually that I have conducted three from my undergraduate up to now. I have conducted six research project, but I then as a zoology special graduate, I need to actually I like to step into that molecular side, but I have some much struggling with inside me that whether that only I have some much basic knowledge and the background knowledge for the molecular side now, then I need to ask whether I will step up in my postgraduate with the molecular biology or the biomedical sciences. What are the preparations of the what are the direct pathways or the what are the specific pathway to step into that medical biomedical science or that subject, because only I have somewhat basic knowledge in my special year and undergraduate. That's the thing. That's why I need to clarify that 5%.
Okay,
Kajan,
thank
you
you,
want to
Dishani.
comment
Yes,
on that? So Dr. Samit has, yeah,
The question is not clear.
she's got zoology
Uh,
background.
yeah, she want to learn molecular biology, right?
That's
Yeah.
what
Yeah, that's good. So better to start with some online courses, so maybe you can learn by yourself. Now there's AI everywhere. You can go to, and also you can search other university websites, and also if you use better keyword searching, you can find the presentations and their lessons and also their practical schedules, these things. I think you should learn how to use the keywords in the Google, so that will bring more impact. So
Yes, sir.
if you want at the beginning of the lesson, I can use how to use the keywords in the Google. So by that you can learn how to learn anything from known to unknown.
Yes, so then actually in my mind from my childhood that I need to design something, create something. So actually that from the molecular biology up to designing designing, I need to somewhat direct path, because in the common scenario we have different sources and different aspects.
Yeah,
No, sir.
for that you should be more specific and more experience in this basic stuff. Then if you are mastering the basic stuff, if combine these things, these basics, you can create a new thing in the interrelated areas. So that's
so,
how you
yeah.
can master those new things that
Yeah,
you
that's
don't know.
something I always discuss with my students as well, Kajan, that if you want to change something in biological systems, you need to know the fundamentals in it. You should know in and out what you're going to change in it. So otherwise you can't blindly apply before you go into applied aspect. So as yeah, Mr. Kajan recommended Dishan, uh, one of the main ways to start exploring any subject area is that uh, thoroughly go through fundamentals. There's a set of books that we can recommend if it's molecular biology. What I recommend is that everyone who wants to start with molecular background should read uh, the molecular biology of the cell book and the genes. And also for biochemistry, Lenin and Voigt and Voigt. So we should go through those fundamentals as well. Meantime, I remember Kajan mentioning as an expert in molecular techniques. Kajan, one of the best ways to explore the tools is that go to these manufacturers' websites and see what sort of video tutorials and certain materials they share with others, right? Let's say you want to learn qPCR
Also, there are
technique.
YouTube channels.
There are YouTube
There is,
channels like so. So because
you
they are
go through the YouTube,
developers.
there's a, so sorry for interruption. So if there's a YouTube channel for this Canadian company called Applied Biological Materials. If you go to that YouTube channel, you can learn the fundamentals of this PCR, qPCR, and also the cell culture techniques, and also this viral vectors. Everything is there, and also NGS. So I think you can go through the channel, Applied Biological Materials, search in the YouTube and go to the channel, and there are more introduction videos that are very simple and also informative and also easy to understand. So if you had a chance, just visit that YouTube channel. Also, I can share that channel's link in this chat as well if you need.
Also, Kajan, we can later share on our Discord platform as well because they are becoming part of our community through Discord platform. So what
Yeah.
areas to explore, right? Dr. Samit, do you want to share some insights as well? Yeah.
Very
We shall move on to the next question, Pabodi. Yeah, you can pick one.
Yes, sir. So the next question is there from Ravina Veerasinghe. She was saying that I'm a fresh graduate from biomedical science. Can I start career in computational biology or bioinformatics? If it is possible, where should I start?
The question goes to Dr. Samit.
Dr. Samit, we can't hear you. I think you're muted.
Okay. So, sorry. First of all, I think you thorough your fundamentals. So, when you come to the computational biology, there are several areas or sections in computational biology. So, even the Dr. Alinkar's domain, the Kajan's domain, and also my domain, like structural under the computational biology, I'm talking about the computational structural biology. So I think first of all, you should thorough the fundamentals about the computational biology fundamentals, and you asking also about the bioinformatics. So the theory is the main thing. When you strong your theoretical knowledge, then we can start the practical things in computational biology. Like you asking about if it is possible, where should I start? So so your first graduate means most probably you are in a first year in your degree program. So when you have the strong or some kind of a level of the theoretical knowledge, you can start some kind of a tutorials kind of things. And also I'm recommending you should learn at least one type of programming language. My recommendation is the Python. Then you can go to C++ and the Fortran or Rust or something like that. But it is very easy to learn Python. So as a fresh graduate, and if you have a ambition to be a computational biologist in future, please start to learn the Python. In these days, you can very easily get Python codes from the AI, even from the ChatGPT. But you should need to set up your foundations about the basic of the Python. You need to learn the basic of the Python. Then you can easily get into the things. And before your undergraduate research, you can do some tutorials like small projects. You can open a GitHub account and do some kind of a small individual project for yourself. Yeah.
So sorry for interrupting. I'm not a
Yeah.
first year student. I'm almost graduated.
Okay.
I'm having a question. As an undergraduate student, I did kind of a research work uh, from a variety of fields like cancer projects, molecular entomology, and recently we did a research project and we are hoping to apply for a patent. It's kind of a integration of AI, electronics. It's kind of a group project. So I'm now
Mhm.
highly interested in AI and the stuff. Now I don't know kind of how to start, but with your answer, now I got an understanding to how to start. I'm thinking the way it's a good because I'm having kind of a research publication for applying for a PhD. It a good for going for computational or bioinformatics. Uh, should I have to look in my field? I'm currently working as a volunteer research assistant at GMB of Chapura. So I want, if it is good, if I stick in that area or go into my interest area.
What is your undergraduate project?
It is a molecular entomology project.
So it's mostly about wet lab.
Yeah.
Okay. So, but you don't, like to continue with the
About
entomology.
mm,
Okay.
like killing animals again.
Okay.
Naveen, would you mind if I ask what's your first degree is BSc program?
So it's biomedical science.
Right, okay. The
And
the
so
Sunny
after
you
that,
can continue on, answer. Yes.
Yeah. Probably you can prepare a small plan if you are going for computational biology, and you should think about what area of computational biology you need to proceed, whether in the structure, computational structural biology, or mostly into the bioinformatics, or maybe synthetic biology. First, you need to decide that. And if you have the theoretical knowledge, you can start like you can first of all. uh, I can speak about my area. If you are going for a postgraduate degree in computational structural biology or computational chemistry or computational biophysics, you should need to have some kind of a knowledge of programming language. Otherwise, it is a little bit difficult to survive. And the thing is, like unfortunately in Sri Lanka, most of the time people are just doing some kind of using some kind of software or online server to get some computational results. That is not the computational, the biology or computational chemistry that the outsiders needed. Most in this AI era, it is very, even for very less experienced computational biologist, or the if we have a more main domain of a lab research, they can use this new kind of AI tools and things and get some computational results very easily. So if you are going for the PhD or masters in foreign country or foreign university, they mostly like to do some kind of a method development, or like software development, method development. They don't need you to just a person like using just a software. So one thing is you should get strong theoretical knowledge, and like learn programming language and do some kind of little projects. Like if you want to go for machine learning, computational drug design related thing, you can do some kind of a toy project, toy models for that, or molecular docking. You can it is at that your level, it is a little bit difficult to develop a method or something like that, but you can use some kind of a the open source softwares and maybe doing the results upload into the GitLab and something like that. And even even if you are try to use some molecular dynamics simulations, you can, it is without support of a HPC system, it is little bit difficult to run more than 100 nanoseconds. So you can run small molecular dynamic simulation from your laptop or desktop with 20 nanoseconds or something like that. And you can have some results and you can present in a local conference, or maybe internet, maybe you can go for some kind of a conference paper, something like that. Yeah. Those things maybe you can do, but be careful about taking a if you need to change your main discipline from your current one to if you need to transfer for this one. So one thing is maybe you can start a master degree and do the master degree research project as a computational one, or maybe MPhil degree with the computational chemistry with a local university. Then you can, after the master or the MPhil, you can go for a PhD in a foreign country in the computational chemistry or biology field. The one tactic you can use something like that. What I want to say mainly about strong your theoretical knowledge and learn at least one programming language and try to do some small project in maybe in a 1 to 3 months, and maybe publish abstract or conference paper, maybe maintain a GitHub with those results, maybe your models, maybe your workflow design, something like that. And I think in future we also can collaborate with you. Yes, how about that, Lalinka? If the
Definitely, Dr. Samit. One of the main ideas of creating this platform as well with Biolum Island is that to expand chances for the undergraduates based in Sri Lanka, right? So like, get them on board. So that's how the idea initially, to get more and more opportunities for our students.
yeah.
They
So
can,
maybe as a
yeah.
part-time project, you can do your main job in the university, Japura, and in the weekends or something like that. We can do some kind of part-time research project. We are here to support you.
Thank you very much for your kind words.
Excuse me, am I
Shall I
audible?
interrupt? Yeah, shall I interrupt? Also, it is now after university education, we expected the graduates to be self-learning more often and not like a school education. It is highly spoon-fed in Sri Lankan education system. So we can only guide what are the tools or how you can do these things, but we cannot spoon-feed everything to you. So that is the most important thing you should understand. Okay, the next question.
Yeah, thank you very much, sir. So I think there's one participant was talking.
Am I audible?
Yeah, you're audible.
Yes, so I'm an undergraduate from University of Pune, Faculty of Agriculture, so I'm in my final year. Actually, in this final year, we have learned bioinformatics and how to develop the polygenetic tree, and we are learning some kind of R language. And I myself, I have done Java a little bit. Um, so after doing that stuff, actually, I'm really interested in this bioinformatics sector. Unfortunately, I choose my final year research from the plant physiology section. But I'm really interested in to join for a research like ongoing research in the bioinformatics sector, like doing the data analyst things and how to sharpen my R language skills and the stuff like that. So I need to know where can I have an exposure, or where can I have the contacts, or where can I have the able to have find the projects which I can enroll with, or which I can use to sharpen my skills with the collaborations. And I need to know where can I be. I mean, the opportunities in my sector, like the stuff like that. So I'm majoring in plant biology, agricultural biology. Yeah.
Yeah, Berneke, right? Yeah. Thank you for your question, Berneke, and for expressing your interest. So, as bioluminescence, what I could say is that we are trying to create a network among Sri Lankan universities, state or non-state expert to link up with our platform throughout our upcoming programs. And then we will also be launching our internship program later. Pavodi will announce. The idea is that to get more and more opportunities for you all to work on certain projects, research projects based on your interests. So, yeah, Sanchula, are you around? So there are students with a lot of inquiries. So what I recommend is that take note on their interest and make a list of students who are interested in different areas, right? So in future we can, if any opportunity comes up based on their interest, we can get them on board and provide them chance to get exposure on different aspects, right? So we are working on that database as well with bioluminescence. You can touch base with us, right? Okay.
Yeah, thank you very much, sir.
And also, it's good to hear that you are doing the R programming language. That's good for the data science projects, and also it is a much needed future-proof skill. So we can continue learning R, and also it is sometimes used in bioinformatics, and also to do some advanced data analysis. And also it could be used to create workflows to solve the complex problems. So go ahead with that R. And also if you can also learn another programming language, and also structured query language, SQL, something like that. So that
Yes,
will
sir.
be very useful.
I did a comprehensive Java programming that I've learned somewhat SQL, but now I kind of forget all those things. But I'm trying to do that again.
Okay, good. Thank you.
Thank you, sir.
Okay, thank you very much, sir. Hope Vanek got a good idea about that, his question. So there's another question from the chat box from Saina. So she was asking, like, I just finished my first year of my biomedical science degree. As for me, this is a completely new and foreign topic. So I would like to know what are the resources I can refer for before joining this session so that I can have an idea about what is this, and is this workshop will be beginner friendly? Do we have to have any kind of background knowledge?
Yes, probably for my session. So basically, the students who are joining, they need the basic knowledge in molecular biology, but still, what I'm going to do is I'm going to simplify as much as I can and let anyone joining can understand the concepts in designing synthetic gene circuits and all. And also in beforehand, I'm going to share a set of materials that they can refer. Maybe by Friday, we can share my materials that are important for them to refer and get an idea. By Friday, I can share them with the students who are joining. They can get a basic idea about synthetic biology, starting from how actually molecular biology evolved, like synthetic biology was starting from fundamentals of molecular biology, and then came the recombinant DNA technology. After that, it expanded so into the genetic engineering era. Now we are in synthetic biology era, where we design life entirely from the scratch. So we pretty much need to apply the engineering concepts that comes from maybe electronic engineering and also the other engineering aspect like DBTS cycle. So these are some topics learning engineering topics. But we are trying to integrate under synthetic biology platform all these aspects with biological fundamentals, biological science fundamentals. So I will share some fundamental to refer as well before they join with my session on Sunday. So I can have them shared by Friday. But don't worry, anybody who's joining, I put my effort to make sure that you'll understand the basic of synthetic biology into our session, and then as Kajan mentioned as well, the idea is for later for you all to start exploring from the basic knowledge that you get through our session and then start exploring advanced tools. So we can also guide you throughout the journey of which tools to access because we are not going to give up on you. Once you are done with the workshop, we have the Discord platform that you guys can keep asking more and more questions even after the workshops as well. So yeah, so I encourage everyone to join me. Like students in first year, second years, third years, because if you start going through these areas, maybe starting from first year as well, then they have more time to master, starting from the fundamentals and then to master the applied aspects of it, right? So doesn't matter you are in first year, second year, third year, or in the final year. So you are not late to start exploring a new area. Of course, it's new and it's keep evolving as well. The knowledge in molecular biology is rapidly evolving, everybody. So we have to stay up to date. Otherwise, we won't be able to keep up with the global knowledge. It's all about reading, going through the materials, and trying to learn new tools. So that's the way to go in synthetic biology as well. It will evolve into another phase as well very soon, the synthetic biology. So we should be keep, it's a learning curve. Yeah. Okay. Right. Any other opinion?
Okay, so for my section, it's highly based on Python, but you don't need to have a knowledge of Python. I mean, for this session, because we will provide you the Jupyter notebooks and the other resources. And also, I think we already send you the preparation guide. For even for that preparation guide, if you have any.
Sorry to interrupt before I forget. So about specific tool in my session as well, there are two tools that you'll be using, which is SBOL Designer tool and iBiosim interface. So to run iBiosim, you need Java runtime environment to be installed on your computer. So that guideline I will share by Friday. SBOL Designer, it's a web-based platform and the databases as well you can access. So that's the specific guideline. I will prepare a document and share it by Friday with all of you. Okay. Sorry, Dr. Samit, again.
Yeah, you no need to know about Python. We will provide you the Jupyter notebooks and all the codes and the other resources. And also, we already send you the preparation guide. Even for preparation guide, if you have any doubt or any technical difficulty, please send a message or just a post on the Discord with your screenshots and anything. We will help you. And I think, Lalinka, we will in the future we will provide some kind of a Python for biologist or Python for biomedical
Of
research.
course, of course. Yeah, we have a
Course.
plan to launch a short course on that as well. So it will be run by
Yeah,
Dr.
it's
Samit.
nice to you have some kind of knowledge of Python, but it is not essential for this one. We will provide all the Python codes and the automation workflow, and in the workshop I will explain how these things are work. Yeah.
Okay, thank you very much, sir. I think, Sainav, you got a good idea and you clarify your question. So how you should prepare before the workshop. So, and then we have another question from Jasmine. Hi, I'm currently doing the biotechnology degree as a third year student. I'm only know about bioinformatics. This area is completely new field for me. I do not have more knowledge about Python, and I went through the guidelines. Some keywords are also new to me, as I have less knowledge about this, but have great passion for computational biology. When in this field, how I gain more qualification in this field?
So I think I also mentioned in the previously that if you are very new to the computational biology and the Python and the related things, you can do some kind of a small the personal projects, some like things, toy models, and the follow some tutorials. And in the future, as the Dr. Lalinka mentioned, we will provide you some kind of we will the courses about the Python for biology, Python for biomedical research, something like that. So even for those courses and workshops, you can get some kind of knowledge, experience, hands-on experience about how to familiar with these things. So after these kind of a tutorials, workshops, then when you at the final year research project, you can do some kind of a computational biology related the undergraduate research. It not need to be the solely or the mainly about the computational biology undergraduate research project. Maybe it's a part of your project, like you can have some wet lab content and some kind of a little bit part like the if you're working with a protein or something like that, you can the model the protein, you can characterize the protein using computational biology, something like that. From those kind of things, you can add some the things to the profile, your career profile, your CV. You can add those kind of things to, you can show that the world that you have some experience, knowledge about computational biology, the domain.
Yes, thank you very much, sir. And if our participants have more questions, you have some time, so you can ask any question, or do you have any problem to clarify? You can use this time to ask from our speakers. So I think
Yeah.
yes, sir.
I have a direct message, but from Sasini about the bioinformatics. It's about immunoinformatics tools. Probably this question may be answered can answer by Kajan. Sasini, can
Okay.
you? Yeah, Sasini,
Ask
can
the
you
question?
share the question in the main chat? Then the Kajan can answer this one.
Okay, sure, sir. So, good evening, sir. I'm actually a bioinformatics postgraduate student. So, first of all, I should tell that I'm from a microbiology background, and also now what I learned from all three of you, it was really interesting. And for the fresh graduate students who is there in the uh, discussion, I should tell you, like, if you really have the passion on computational biology, now since I'm a postgraduate student in bioinformatics, it's not a barrier. So as all these three expertise told you, you prefer like get on skills on Python, R, and especially like the basics, fundamentals on molecular biology as well. So eventually, even though you're the basic, the fundamental is some like different field, maybe not molecular biology, but maybe like I'm a microbiologist. So you will eventually, you can work on it, but there will be, as mentioned, there will be so much of hard work, like which you need to actually put on. So now what I was like thinking about now, since I'm working on my the postgraduate research, I'm looking for self-studying things which is actually needed, like because we know the theory but the application part is what is missing, you know, when we studying the theory part. So that's very important. While you are learning the basics, getting know how to apply it, it's the really important thing when it comes to research. So at the end, that's what every one of us is expecting. So actually I want to know, sir, that whether there is any specific websites or YouTube channels that is related to immunoinformatics, because my research is working on that, because there are many informatic tools that we need to refer when it comes to vaccine construction, like within the workflow. So it will be different software. So it's really hard to like learn each and every one. So like every tool. So any specific website or in like known YouTube channel to get to idea about those tools.
Kajan, maybe you can
You're
talk about.
especially focused on immunoinformatics, right?
Yeah, yes, yeah.
So deviating from the microbiome, these things, microbial genetics. So for the immunogenetics, we saw something in this website called that is a Genius. That is a software provider that's from New Zealand. So Genius Prime, there are tools for this immunogenetics. But it's a paid software. You can get a student license for around $200 per year or something. You can try that software.
And
Okay.
also, Sasini, have you been working on chipseq analysis project? Is it related to NGS analysis, your current project?
It's not like NGS. I think I worked on for the MAFTA analysis. So then like to the conserved regions identification. So afterwards on like there are different other tools like epitope predictions and stuff. So for that, like many tools are there, but yet like I'm struggling on to find out how those things are specifically working on. So I was not able to find out like separate softwares. I mean, as one place where I can learn on everything. So that's what I was looking. So.
Genius
So
is a
there
good
is,
recommendation. You're welcome.
Okay.
Sashini, there is no single website.
Okay.
So there are tools, and also if you want to learn something, there are now it is broadening the NGS area. There is metagenomics and also cancer genetics, and also these epigenetics things, and also
Yeah.
the single cell things, the RNA-seq, chip-seq, and
Chipseq,
also and also now it is we are going to entering in the era of proteomics.
Yeah. So it's mainly on epitope prediction. So
Yep.
like into a multi-epitope vaccine
Yeah,
construction.
try that
Sami,
software called
Dr.
Genius
Sami,
Prime.
Okay.
Yes.
yeah, work on epitome analysis and all, you can recommend something, right? Predicting epitome sense.
Uh,
That's on immune. like to predict protein segments, the antigen segments.
Okay, so this is about the protein segments.
Yeah.
Yes, sir.
Okay, so you can try AlphaFold, but I'm not sure about whether it can accurately predict the epitone because I never used AlphaFold or other new generation of AI tools to predict something like epitone. But you can try, and before this new generation of protein predictor and AI tools, the previous methods are basically about the homology modeling, and probably the homology modeling like modeler maybe you can use. But first of all, I think you can, if you need the structure, and if this is a part of a protein, something like that, you can use the alpha fold, ESM fold, omega fold, and pep fold. I can't guarantee how the accuracy of these things for the epitone, something like that, because I already worked with the peptides, and there are some issues even for the peptides because peptides are small protein, less than 100 amino acids.
Yeah.
So they are not perfect, but AlphaFold is considerably okay to peptide, but it is not accurately predicting the peptide if they have the sulfur bonds or something like that. Sometimes there are some issues even for peptides. You can try AlphaFold, ESM fold, omega fold, and pep fold. And probably you can try the modeler like homology modeling for this one also.
Maybe it must have been trained, Dr. Samit, as per my understanding, because the immune proteins are also widely annotated. The AlphaFold tool. At the same time, Sasini, what I recommend is that to use multiple tools to verify your outcomes, as Dr. Samit mentioned. So you
Okay.
can go for the homology modeling as well as use AI tools. Yeah.
Did you check with the some kind of a publication, previous publications?
Yeah, sir. So like few publications they have mentioned about the AlphaFold. So but I need to work on like how it like actually work now. Yet I have not into the software. That's the thing now. Though like they have mentioned the steps and all, like learning about the softwares, that's the part that I'm really struggling. That's why I was like looking any available like a course or something which is there, which is related to me.
Dr.
So,
Samit, we haven't announced it, right? We have already planned a small workshop on AlphaFold.
yeah, the next, most probably the next workshop is about AlphaFold, ESM fold, omega fold, and pep fold related workshop, how you can use these AI tools to predict the proteins. Also, what is the theory behind that, and what kind of a mini project you can do using these the protein prediction tools. So still, it's a the bioloom still not announced that, but most probably the next one is about the protein prediction.
As it came into the topic, so we will be announcing it very soon, Dr. Samit. So the workshop on AlphaFold just after this, most probably in January. In January, we can have a session on AlphaFold.
Yeah, Sasini, probably you can first of all try AlphaFold, and then also simultaneously you can try the modeler. There is extension in the Chimera for modeler. if you need some kind of help, probably you can contact me via the what is called the Discord, or maybe you can email me. I I, I will try to give some help what I can.
Okay, so thank you so much, sir.
Sure.
Okay, thank you very much. So do we have any question from our participants? I think you got a great idea and you clarified a lot of questions. So any other question before we end? Yes, sir.
Yeah, there's a direct message from Zainab. I will answer for that in the public. I think it's okay. She asked, thank you so much for the clarifying, and was concerned that this might be a too advanced. I appreciate your clarifying. Yes, this is for, I can guarantee this is not an advanced one, especially in my the section. Yeah. Okay, so this is a very first steps of the computer-aided drug designing workflow. What we are doing is in the upcoming workshop is we are just get the main information of the structural information from the PDB and UniProt into these first few steps of the computer drug design actual workflow, and this is like a open source, and this is automated one, so not only for one protein or any biological molecule, you can apply for this one for the entire set of the biological molecules, proteins, and other things. And you can tailor this one because this is open source and Python. Even when after this we provide the codes and things, you can change the code, anything. And we will most probably, I will provide the GitHub. Then you can fork this workflow and whatever you can do as you decide. It's okay. This is a so that is the main benefit of this. If you are using like a ready-made software, you don't know what is happening there. You can change the things. There are very limited options you can do. So, and this is the kind of a very good experience to learn how the actual things are work in scientific community, how the open source workflow are working. Okay, so I can guarantee that this is not an advanced one. Even we are providing all the codes and all the technical support here.
Okay, thank you very much, sir. So do we have any other questions, dear participants? So this is your chance to ask any question that you want to clarify before the workshop. So all our three speakers are there and they are waiting to answer you. You will have a few more minutes if you have any question to clarify.
Good evening, sir. I am a finally a BPharm undergraduate of University of Peradeniya. I am doing a computational based informatics research on epitope prediction related to vaccine design. Is there any special standardized workflow related to that research?
Is this about epitope prediction?
Epitope prediction related to dengue virus. It's from NCP virus sequence retrieval and multiple sequence alignment, and then IBD website to use prediction of bit of bits. My one is related only T cell a bit of prediction.
If you are, like, as I mentioned to Sasini, if you are predicting something related to protein, you can try it out these new generation of AI tools like AlphaFold, the ESMFold, OmegaFold, or PepFold, and every day there is a new tool they are developing, and another thing I just need to mention, AlphaFold 3, there are some restriction for commercial use. I think in a one day you can only predict maybe 20 or 15 structures, but the AlphaFold 2 is totally open source. You can use that. So maybe you can compare with the if you have the known protein structure and with the experimental results, if you know the data, you can compare with the experimental results with the predicted structure. Then you can like get some idea about how accurate this is. Otherwise, you can use the modeler like homology modeling things for predicting these the small part of these the proteins. So in modeler you can use these the multiple alignment sequence modeling. Yeah. Otherwise, I'm not much familiar with the specifically for epitope.
Pretty much, Dr. Samit. So these are the small protein component that are recognized by antibodies when it comes to immunology. Yeah, so antigens components recognized by antibody. So what I would say is that, of course, you can fold with because these are mostly proteins. Yeah.
Yeah, is it about totally about amino acids?
Yeah.
Okay. So then you can try these AI tools. And if you have the known structure, you can compare that one. So first of all, you can search on the RSCB protein data bank and get some verified this epitope protein structure and try to predict from these tools and also from the modeler. Chimera has a modeler, the extension. You can use that. Then you can get some kind of a idea about how these things, how these AI the tools work on this epitope. Not a one single one. Maybe you can try it with the 50 or 100 if they are available, and you can do some kind of a benchmark work. And after the like when you compare, you can make a workflow. What kind of characteristics you can compare these two. So if you have a knowledge of molecular dynamic simulation, you can extend this into the predictor structure, run into molecular dynamic simulations also, and then the same available structure run into the molecular dynamic simulation and do some kind of analysis part using this molecular dynamic trajectory. So it's a little bit big part, but basically you can do like download the available epitopes and predict the epitope from different AI tools and compare those sticks in structural manner.
Okay, sir. Thank you.
And do we have any other question? So before we wind up the session, if you have any other clarification details, so here your chance. So if you have any other question before we wind up, you can ask, or else our speakers, Dr. Lalinka, Dr. Samit, or Mr. Kajan, do you have anything to say to our participants?
Dr. Samit, you can go first. Yeah.
Yeah. Please follow the preparation guide. That is main thing I need to say, and do those needed preparation, and we will provide that Jupyter notebook and the GitHub link on probably in Saturday, and then we can meet on Sunday at 8 a.m. Sri Lankan time.
Kajal.
So
And sorry, another thing, if you have, again, I'm saying, if you have any technical or any concern regarding the preparation, please send a message via in the Discord message or any post. We will here to help you.
So, so regarding the my sessions, so if you visit on my website, it's hosted in snp.biotech.edu.lk. You can find the tool what I am going to explain to you all. And also you can find the publications related to in the hyperlink given below. So you can go through the abstract. It's a subscription journal. You can get the whole paper. Only one paper about a tool, you can get the complete paper. You can read the paper and get prepared to learn about the mutations and how to identify the low cost setting. So thank you for joining me.
And yeah, okay, Kajal. Yeah, for my session as well, technically what I will do is I'll share a specific guideline for you all to utilize SBOL designer tool and iBiosim software for simulation. Specifically for iBiosim software, I will have a kind of lengthy document that you can follow as a stepwise guide how to utilize iBiosim simulation tool as well. So I can have that document shared with you as well by Friday, everybody. So that's about technical aspect. Yes, and for the basics to understand, as some students were worried about their fundamentals in molecular biology, I would also have shared a few recommendations for you all to go through and glance and come before you join my session as well. Yeah, that's about it. Yes, Pavodi, over to you. You can wind up and announce about remind them about our internship program as well. Right, okay. Thank you very much. Yeah.
Yes, thank you very much, sir. So all our speakers, Dr. Lalinka Herat, Dr. Samit Ratnayaka, and Mr. Kajan Muneeswaran. Thank you very much for this comprehensive idea. Before we joined in the workshop, you gave a good idea to our participants. So what they have to prepare before the workshop and what they will gain through the workshop. So, and there is a special notification. So throughout this Q&A session, you observe that we have shown you the promotional code for the discounted prices for the workshop. So by using that promotional code, you can register for the workshop by paying 6,500 rupees. So you can use that promotional code as well. So as Dr. Lalinka mentioned about the internship in the Balum Island, so I must mention that also. So we are happy to announce that the participants of this workshop will consider for the internship opportunities of the Biolum Islands. So the internship will be mainly focused on the synthetic biology projects, in bioinformatic tools and softwares, molecular diagnostics, data-driven biological researchers, wet lab, and computational hybrid projects. So if you got this internship opportunity, you will get the exposure to real-world R&D and scientific communication and publishing as well, and also the industrial workflow, and you will get the chance to collaborate with the researchers working in agriculture, biotechnology, and also in health innovation as well. Because the Biolum, we believe, the empowering young scientists with hands-on experience is that can directly support their future research and employment, and also postgraduate studies. So with that, I think it's great time to wind up this session, because all our participants got a great idea about the upcoming workshop, and you clarified all of your questions. So with that, we can wind up the session. So thank you all for being a part of today's meet and greet session. We are excited to see you for the full workshop, and recording guidelines and support resources will be provided throughout email and also our Discord community. So with that, we will wind up this session. So thank you all. Have a nice day.